Good morning, everyone. I think we can get started. So again, good morning, everyone. It is my greatest pleasure to welcome all of you here in person and those joining us virtually. This is Wei Gao here. I'm from EMD Serono from the Department of Quantitative Pharmacology. I'm here to represent the ISOP, International Society of Pharmacometrics. I'm thrilled to open today's insightful workshop. A few very quick housekeeping items before we begin. So as you can see, coffee is at the back of the room for our guests here. Lunch will be served across the hall. During each session, our moderators will briefly introduce the speakers and panelists. For the speakers and panelists, you will sit here together with us, and for all the audience, please save all the questions for the end. We will have a Q&A and panel discussion at the end, where both our in-person and virtual participants can engage with our panelists. Okay. Now let's set the stage for today. This workshop is a collaborative effort between the FDA and ISOP. It will focus on the dosage optimization for oncology in combination therapy and for new indications. The graphs here show the trend of oncology product approvals over the last 20 years by the FDA, contrasting single agent in blue and combination in red. The complexity is underscored by the plot to your right, mapping out the web of 187 combination pairings. Each line represents a unique combination pairing. So I hope from this graph you can already appreciate the complexities. So the questions to us would be, how do we select and optimize dosages for combinations? What are the considerations when pairing novel approved or some other agents, or when when we are dealing with the different therapeutic modalities? What are the considerations? I think those are the questions in our mind all the time. Additionally, many oncology agents are being investigated for multiple indications. The graph here just shows the landscape of indications that certain PD-1 and PD-L1 products are pursuing right now. So the same pressing question came to our mind. How do we select the dose, and how do we optimize the dosage across such a broad spectrum of indications? What bridging strategies can we employ? Identifying and addressing knowledge and evidence gap is also very important. That is a task that we have to undertake collectively as a community. So the FDA's commitment and dedications to the rational oncology dosage optimization is evident under Project Optimus. The regulatory framework has been enriched with guidance development, MIDD pilot program, multiple presentations that we have heard from FDA. And we oftentimes heard the encouragement to have the direct dialogue with them through those interaction meetings, including the Type D meetings. And the recently issued draft guidance on oncology dose optimization, as well as the recently launched dosing selection toolkit are some of the very recent efforts. So I just want to thank Dr. Stacy Short, who's the co-organizer of this workshop, for providing this slide. Okay, so as I previously stated in the October 16th workshop, that was the first workshop that ISOP and FDA has been co-sponsoring this year. I use the quote here. The oncology dosage optimization is a multi-dimensional problem that demands a totality of evidence approach reinforced by a MIDD mindset. I think this perspective aligns seamlessly with today's workshop as well. At the ISOP, we are committed to advancing pharmacometrics, which is pivotal for rational dosage selection and optimization. Our collaboration with the FDA is a testament to our shared goals of using quantitative approaches to enable rational oncology dose selection for combination and new indication, as you can hear here. And now I think it's also the right moment for us to have those dialogue as we as a community are embracing the paradigm shift of oncology dosage optimization and drug development on the Project Optimus. So today's workshop has 3 sessions. So first one, we're gonna outline some of the understanding of regulation and impact of response variation on dosage selection. And for the second and the third session is dedicated to the considerations for the new combination and the new indication correspondingly. Especially our moderators, speakers, and panelists will highlight the need to select the dosage for new combination and new indication using totality of evidence. We will discuss the factors to be considered when moving into a combination or new indication. And you will hear how model-informed and model-based approaches can be used to select dosages for new combination and indications to be investigated in clinical trials. Without further ado, I would like to invite the moderator for the first session, Dr. Hao Zhu, to the stage.
Thank you very much, Dr. Gao. So it's my pleasure to open up the first section of the workshop. In this section, we're going to focus on 2 topics. Number 1 is the regulation, and number 2 is what are the challenges we see for oncology dose finding. This section includes 2 presentations and followed by a Q&A section. The first presentation will be presented by Dr. Chow, who is an associate professor from School of Pharmacy in the University of North Carolina and Chapel Hill. So let's welcome Dr. Chow.
Thank you for the very nice introduction. Let me see whether I can control the slide. Know how to control the slide?
Okay. Can we go back to the earlier slide?
All right, so it's truly a great pleasure to be able to speak in this great meeting organized by ISOP and FDA. Today, I'm going to take about 20 minutes, share a little bit about our experience using model simulation, try to understand the response variation in the oncology drug treatments, and to see whether our knowledge learned throughout the process have any implication to the dose selection in the drug development. Let's get to it. All right. So tumor heterogeneity, I think I don't need to say anything to this group of people. Probably is one of the most challenging, if not the most challenging aspects in oncology drug development. That can lead to lead us to 2 challenges in developing the oncology drug. One is inadequate control. If you think about when you recruit patients, the patient cover a whole spectrum of variation in terms of the characteristics. This is not a consistent or homogeneous population. This is a population with a very heterogeneous background. Sometimes compare the treatment arm and the control arm can be challenging, particularly if you want to break down the treatment arm into subgroup, comparing that control, that's going to give you a challenge, lead to an inadequate control. Another point I want to make that if there's a heterogeneous population, right, the population average will not well reflect the individual patients. If you have narrow defined population, the population average probably can represent a large fraction of the population. However, if you have a broadly distributed population, the population average only reflects a fraction of the individuals. So how to overcome the heterogeneity? Heterogeneity exists at multiple levels. I think the drug combination probably so far is the most effective method for us to overcome the heterogeneity. So the heterogeneity can present at a patient level. Some patient respond to drug A, some patient respond to drug B. If we combine drug A and drug B, then we can reduce the chance. Now the patient going to respond to the treatment, right? That's the treatment level. At the patient levels of heterogeneity, we can potentially increase the overall response rate. If the heterogeneity present at the cellular or the tumor cell level, then we can, increase the chance of the response of the tumor. Some cell is not responding drug A, some cell not responding drug B. Then we combine them together, then going to have a higher chance to shrink the tumor. There's a chance we can improve the waterfall plots, also mean the best response. If the reduction of the tumor size getting deeper, then we have a chance to reach a complete response or curability of this treatment. So there's many ways we can integrate the evidence or knowledge throughout the drug development. So we call this a totality of evidence. We can use the preclinical evidence, we can target engagement, PD markers, dose response relationship, or dose toxicity relationship. All those analyses, at the end of the day, we want to evaluate their dose dependency to see the response depend on the dose or the toxicity depend on the dose. Eventually, we want to select a dose, balance the two factors the patient can tolerate. Meanwhile, we can reach nearly maximize the efficacy. So this is a simulation study based on approved drug. Without naming the name, you can see that on the population level, the dose response is quite clear. The 90 milligram is better than the 40. There are about 25% response increase at the higher dose compared to the relatively lower dose. That's the population level. However, if you break this down into the individual level to draw an individual conditional expectation plot, the expectation plot is quite simple. Using the model you developed, the covariate you identified, fix the individual covariate, simulate the dose response relationship within that specific individual. You can see this relationship not always in line with the population levels of dose response relationship. In some of the patients, particularly the patient on the top, about 50% of the patient, 40 milligram already okay, nearly reached the saturable level. 90 milligram, it's not giving them additional benefit. That's for 50% of the patient. That patient tend to be the patient with a mild disease status. We identify most of the patient have the ECOG equal 0. The patient on the bottom, that's the patient very severe disease condition and ECOG 1 or higher. So that if you choose a 90 milligram, if the tolerability is not the issue, that should be fine on the population level. No problem about that. However, if you want to do a early stage dose comparison study with a relatively small sample size, there's a chance, even with randomization, there's a chance you can just select the patient in the 40 milligram more favorable to the response. There's a chance about that. We should consider those sources of the variability, understanding the factors contributing to the response so that we can have a more confidence dose response relationship. Another example being conducted by Genentech, I think lots of the senior key players on this project sitting in the audience. So this is a TDM1 study. If you just look at the population comparison to the control without breakdown, just the group control, break down the treatment group into multiple subgroup, Q1, Q2, based on the exposure. The Q1, which is the patient have the lowest exposure, just look at the left panel, is worse than the control, indicating there might be a detrimental effect. However, if you look further, the control is not is not adequate because the control is very heterogeneous. If you tease out the patient not matching up with the Q1 patient, look at this dose response again, then adjusted Hazard ratio will be lower than 1. This is the indication to us that we should think about all the factors contribute to the dose response relationship so that we can be more confident to get a more clean dose response relationship. So if you look at the literature, similar study has been done many times, try to tease out all the confounding factors to give us a clean dose response relation. So the point I want to highlight here, if you want to join the DR relationship on the population level, that's largely is the correlation analysis, not the causality analysis, because you're not quite sure whether the dose is a factor leading to the response getting higher or lower, right? That's a correlation, not causality. We should perform more causality analysis to understand that better. So to choose a dose, if you really want to make sure the dose you choose is the reason we get a higher or lower response, we should perform the caudal DR relationship analysis. In other words, it's a clean DR relationship. So what are those parameters? What are those factors influencing their relationship? If you look at the whole cascade of pharmacological action, the factors can potentially influence the dose response, can break down into many aspects: the patient characteristics, the tumor characteristics, the gene profile, the clinical pathological factors. All those factors can potentially influence the relationship between the dose and the response. We want to look at their factors, compare them across scale to see which factor influence the most. This is a simulation study done by our group, using a HER2 positive breast cancer as a system. So in building this framework, we consider many aspects of those confounding factors, including the pharmacokinetics, pathological factors like a HER2 IHC score, tumor characteristics, the growth rate, and such, and also some drug potency factor being considered. All those factors being integrated into a unified framework. The whole point is try to compare what the factor contribute the most in this special case. So before we jump into the simulation part, we calibrate this framework using the progression-free survival curve, reported by the two drugs, neratinib and lecanemab. You can see that our model can roughly, I'm not saying fully characterize, fully calibrate, but this model roughly can reproduce the PFS curve. So the first factor we evaluate is the tumor growth. In our model, we consider a resistant clone, sensitive clone, similar to the tumor growth inhibition model, considering the fractions of resistance, right? We evaluate what's the growth rate the resistant clone can contribute to the PFS. Unsurprisingly, this is if the tumor resistant clone have a faster growth, then the patient survival will be worse compared to the patient who has a relatively lower resistant clone growth. If you look at the exposure variation, all those factors being categorized into a multiple subgroup, you can see the Q1, Q2, Q3, Q4. For neratinib, maybe there are slight influence by the exposure around the therapeutic dose, but lecanemab, there's no signal being indicate that the exposure is an important factor influencing the PFS in this case study. So if you compare all those factors being considered in the framework, ranking them to see which one contribute the most, you can see the top three, now of them are exposure related. I'm not saying that the dose is not important in this case. I'm just saying that exposure around the therapeutic dose, the variation around the therapeutic dose, the exposure variation is not a significant correlated compared to the other factors. When I say the other factors, most of them are related to the tumor characteristics rather than the exposure itself. So if you want to find the dose, with that being characterized, if you want to find the dose, we should better understand what the other factors are, right? This is a theoretical analysis. Some of the factors not even being characterized in the clinical study, but I just want to point out that understanding those other factors is critical. Otherwise, finding the optimal dose, to me, is pretty much like finding a needle in a haystack. So randomization is great, can reduce the systematic levels of bias, but not guarantee group balance, right? As difference always happen by chance, particularly in the small trial, in the early phase, those comparison trials, the randomization is not guarantee well-balanced groups. That's the reason why we need to understand those factors. What is contributing factors leading to a bias, dose response relationship. So what we do when we see a very noisy control compared to our treatment, typically we do the adjustment, like the earlier TDM1 case I demonstrate. We adjust those confounders. We're using some algorithm to predict what's the most important contributor, and then integrate them together to calculate some propensity score matching the patient between the two groups or weighting those score between these two groups, then eventually we can have a fair comparison. What's the real dose dependency here? However, like I said, limit early stage data restrict our ability to finding those factors, particularly for the novel drug treatments. You don't know what's the significant factor. You need a lot of data to identify those factors drastically influence the response or the toxicity. We don't know. That's the reason why I push the causal ER relationship to a Bayesian approach. We should rely on those Bayesian approach, knowing the prior knowledge, prior experience, incorporate that into our DR relationship so that we can get some more confidence in analysis. So what are those factors? Again, revisit the cascades of pharmacological action. So in roughly, we can categorize them into two groups. The handler is essentially driven by those two aspects of factors. One is the prognostic, another one is the predictive. The prognostic is the disease-related. We also call this a risk factor in some other area of the health science. The treatment factor, essentially we call this a predictive factors. They are related to the treatment effect, right? At least some examples there, but there's no black and white here. There's lots of overlapping here. Predictive factors can sometimes overlapping with the prognostic factors. So if you look at those two factors, prognostic is a disease-related. They influence the relative, they influence the absolute risk ratio equally between the treatment and control, right? If you have a prognostic factor present, then they influence the risk equally between the two arms. However, predict factors only influence the treatment rather than the control. So that's the relative risk factor can increase because the predict factors. So when I'm saying predict factors or prognostic factors, they are not limited to efficacy. Toxicity is also another aspect we should learn more about this process. This is not a target therapy situation. When you see this chart, this is a small chart, apologize for that. You can see that this is a scoring system. The clinician develops this chart to give the patient a prior treatment evaluation. What's the risk of the patient suffering from grade 3 or above toxicity? And then they correlated with, between the score and the toxicity people suffering. They find out this so-called chart based on patients' baseline characteristics well characterize the dose, well characterize the toxicity, grade 3 or above. So prognostic factors are largely translatable across studies, but not predict factors because they are only translatable probably within the same type of treatment. If you see those curves, you can this chart, you can see that the PD-L1 pretty much is a predict factors. Before the immunotherapy, PD-L1 never been mentioned for prognosis, right? That's predict factors largely related to the treatment. And on the bottom part, you can see the colorectal cancer, the left or right, that's a very important prognostic factors. Regardless of the treatment, the patient on the right survive worse than the patient on the left. So this is a factor being incorporated into the clinician practice every day. Sometimes in the combination situation, so if let's say the drug A is the background treatment, you want to add on drug B on the top of drug A, then the drug A's predict factors can switch from the predict factor to the prognostic factor. They are not absolutely located in one area. The scenario change, then the position change of these factors. So in the combination, just roughly get to the key point of the meeting. When we are combining novel therapies, then we only know probably prognostic, not much of the predictive. If we combine a novel A with approved B, then the prognostic with B, predict with B, and then both approved, then we know both type of factors. So if you look at the FDA guidance earlier this year, I think in May, FDA CDER issued this document encouraging us to consider these potential covariates. In other words, for the potential confounders, we should use those, leverage the guidance to perform more causal ER analysis or dose response analysis. To do that in the early clinical trial design, particularly when we only have very limited samples, we should, using the Bayesian approach, leverage the power of our prior knowledge, understanding the prognostic factors as well as the predict factors. Both factors can be learned and standardized by mining either the historical RCT data or real-world data. and consensus building process is still largely needed. So there are still some open questions around this topic. We are still in the process of exploring. So a couple of things I want to point out. The prognostic factors often differ across different readouts. If you want to see the patient survival as the outcome, you may get different prognostic factors compared to the other clinical readouts. And the risk factors for toxicity remain largely unexplored, particularly for the target therapy we typically discuss. The method discerning or differentiating the prognostic and predict factors are underdetermined. So to make a quick summary, to select the optimal dose, in our opinion, we need a causal DR relationship. In other words, it's a clean DR relationship. The causal DR relationship is quite challenging in early clinical trial studies because we only have limited data, particularly if we are dealing with the novel drugs. We need a Bayesian approach to leverage our previous knowledge about those confounders. And those confounders can further break down into two types of factors. One is the prognostic, another one is predictive. And to learn those confounders, we need to leverage our historical RCT data and our real-world data. We are in the process exploring those things. I think our society can work together, right? We have lots of data to work together to standardize this process, to give us some clue what's the factors we can reach a consensus, which factors can be weighted in the early clinical trial design, in the dose optimization process. That's the message I want to highlight. Special thanks to two students who are working on this project with me, Tim Chi, Vin Dang. Both of them are very talented students. Many thanks to their contribution. Thank you.
Thank you very much, Dr. Cao, for laying out the challenges we see here and provide the potential solutions from your perspective. So now let's move on to our second presentation, where we're going to invite Dr. Thompson and Dr. Shah for sharing their regulatory perspective on non-clinical and clinical considerations on dose selection in oncology setting for both combination therapy and new indication. So Dr. Thompson and Dr. Shah are clinical and non-clinical and clinical leads from the FDA. So welcome, Dr. Thompson and Dr. Shah.
So good morning. My name is Matthew Thompson. I'm a pharmacologist at FDA in the Division of Hematology, Oncology, Toxicology. So we're pharm tox, or the non-clinical reviewing branch. So here are disclosures. And this is a rapidly evolving field. So especially from the non-clinical side, and we'll comment more on that. So what non-clinical studies are warranted to support combination therapies? We could think about it in terms of novel, approved, and approved, and then to support new indications. So whether or not you're moving from a lot of submissions will come in with an advanced cancer indication, if you will, and they are trying to figure out where their drug is going to have activity. The non-clinical package to support that kind of approach as it moves towards a more specific indication, or that's something to consider. You could also consider a new indication could be from an approved indication to and then a newly proposed indication, or moving from a later line to an early line. And in general, the non-clinical studies that were supportive of the initial development of the drug, of the product, are going to continue to be supportive of new indications. So what's our guiding principles? They're going to be laid out in FDA guidances S9, so the safety guidance S9. And then there are additional Q&A. It's an additional Q&A document, the S9 Q&A, that's going to lay out additional points to consider that clarified issues raised in S9. And so the scope of S9 is the treatment of cancer patients, and it covers a range of products, including ADCs and bispecifics, small molecules, etc. So this is basically boils down S9 and the S9 Q&A into one slide. In particular application to this idea of combinations and new indications. So we're going to expect that a non-clinical package is going to have data to support the rationale. So we're interested in mechanistic data showing activity. It could be in vitro, in vivo, or literature-based. We want to see that the individual agents have been studied in toxicology studies. So in general, that's going to be your typical development plan would be 4-week repeat dose toxicology studies in 2 species to support initial, like first in human development for a product. And as long as those studies have been conducted for each agent, then toxicology studies for combinations are generally not going to be warranted. And if you have a human tox profile that's not been well characterized, so it's very early in development, maybe you've only had, you haven't begun your first in human study, or you're so early that you maybe haven't completed a cycle, then you may need to consider a pharmacology study with toxicology endpoints. So in that case, you would want to be demonstrating an increased activity with the combination in the absence of any new emergent toxicity. And in those kinds of studies where it's a pharm study with tox endpoints, we're not considering the entire battery of things you normally see in a GLP repeat dose tox study. You'd be more interested in things like mortality and clinical observations and things like body weight. And then this is a general statement. If there's insufficient data overall, clinical or non-clinical, to establish a starting dose for the combination, then you might need to provide additional information in the non-clinical space. So non-clinical considerations for the use of combination therapies. So in terms of the mechanistic rationale, you could think about it in terms of, there's a lot of aspects we could think of, but the more mechanistic you approach this, the better. And I think that there's a lot of non-clinical data that is developed in either in vitro or in vivo systems. And the idea of using models to integrate that data is very appealing. Also to consider is whether or not therapies are going to be systemic in terms of the application, or they're going to be focused on maybe a localized disease. So thinking about, if, for example, the route of administration, are you going to be using an intravenous dosing route or intratumoral? You might consider additional things like treatment modality. So is the combination going to be involving radiation or chemotherapy or a targeted therapy? So considering that you have provided a strong mechanistic rationale for why you're proposing the combination, the pharmacology should be presented in an integrated way along with the toxicology data. And again, the toxicology data should give us a sense for the dose toxicity relationship. So was there a lot of toxicity or severe toxicity observed for an individual agent, which would raise concern for us? In addition to thinking about severity, we'd also want to consider and the dose toxicity curve. We want to consider whether or not there's additional development in the clinical space. So in terms of if there's emerging literature for a combination that's supportive, or in vitro or in vivo data. And finally, in terms of benefit risk, we want to consider the totality of the non-clinical data and things like safety margins in the toxicology studies and what the pharmacology evidence is supporting. So for new indications, it's the same approach as for combinations. New toxicology studies are generally not warranted to support a new indication, but do present the rationale considering all of the available data. And in particular, this is important if you're proposing a new indication and the tumor biology is different, then we need to be considering if there is a need for a different kind of mechanistic rationale. So different proof of concept potentially, but at least a demonstration of activity and an addressing of potential dosing issues. So here's a simple scenario. So we have a preliminary understanding of dose exposure response relationship, which has, we understand the anti-tumor activity in the non-clinical setting, and we have a safety profile, so well characterized individual agents. And you've developed a model that integrates all this information, which right now is not a very common thing to see in an application, something that considers both the pharmacology and the toxicology in an integrated way, at least from the non-clinical perspective. And so you submit this data and the model to the agency for feedback. So from the non-clinical perspective, this is absolutely an emerging area in terms of we have, for first-in-human dose selection, we have a lot of models coming into the agency to support first-in-human dose selection. So it'll be interesting to see how this evolves in the non-clinical space in terms of combinations and new indications. So consider when you're submitting to the agency that the typical pharm tox reviewer is not going to have the same kind of modeling background that maybe somebody in ClinPharm or Pharmacometrics is going to have. So provide a very clear summary of your model, a tabulation of the data, the outputs, and then state your modeling assumptions very clearly, and particularly why you made those assumptions. And then just consider your audience, consider who you're presenting the model to, explain why you did what you did, and the strengths and limitations of the model. And that will aid the review of the application. Now, Dr. Shah will comment on the clinical considerations.
Hi, everyone. Good morning. For those who don't know me, my name is Marat Shah. I'm a medical oncologist and specializing in breast and gynecologic malignancies at FDA. Sorry, the microphone's a little tall for me. I'm just going to adjust this. And I also co-lead Project Optimus with Dr. Atiq Rahman. I will be presenting my clinical perspective regarding considerations for combination therapies and new indications. And my goal is really to outline some general high-level principles that can be applied throughout the day as we consider the further discussions this morning and this afternoon, and also the case studies. Sorry, how do I advance? So obvious, I missed it. So many of you might be familiar with the oncology dosage optimization draft guidance, and some of the texts in the draft guidance I'm just going to read out loud. I've highlighted some of the key points. So different dosages may be needed in different disease settings or oncologic diseases based on potential differences and various factors related to the tumor, the patient population, the treatment setting, and also if the new indication is for a combination. And there should be a strong rationale for choice of the dosage before initiating a registrational trial, whether that's for the combination, whether that's for a new indication. And over the next few minutes, I'm going to explore these concepts further. So I think, as we all recognize, combination therapies are extremely important to oncology. Some of the potential rationales for developing combinations may include to increase anti-tumor activity, to delay the onset of resistance, to develop a regimen that has activity with manageable toxicity because lower doses of the individual agents can be given. So just some of the many potential rationale for developing combinations. I'm showing two figures on this slide. On the left-hand slide is a bar chart. The orange bars represent the number of trials which supported the FDA approvals between 2017 and 2021. These trials started in 2007, and that's why 2007 is the first year on the x-axis. The line graph in blue shows the proportion of these trials that were for monotherapies, and you can see that the proportion of trials for monotherapies decreased over time, over that period of time. On the right side of the slide, there's a figure which shows the different therapeutic classes that were combined either together or across therapeutic classes to support new combination regimens that were FDA approved between 2011 and 2022 for solid tumors and hematologic malignancies. And together, these figures highlight the ubiquity of combinations in oncology and really the importance of considering principles for dosage optimization of combinations and not just monotherapies. Some of the core principles to think about regarding dosage optimization of combinations, and again, from my clinical perspective, is considering the PK and the impact of intrinsic and extrinsic factors on PK and PD for the individual agents. So important to understand the dose and exposure response relationships for the individual agents. And finally, it's really important to consider how the dosages of the individual agents been optimized. And I'll come back to this point in a few minutes. When thinking about the combination, first I want to talk about some of the high-level general principles for development of the combination before getting to optimization of the dosage. So I think historically, we all know that combinations were developed in oncology in the curative setting, but many of the combinations that are in development today are in the non-curative setting. And inherently, if patients are getting 2 drugs instead of 1, 3 drugs instead of 2, there's a potential for increased toxicity. And something we hear over and over again from patients is, if I'm taking these drugs in combination, particularly with incurable disease, I want to know, do I need these 2 drugs and do I need them at the same time? Or could I take them sequentially and have the same, the same overall effect? So I think these concerns from patients really highlight the importance of having a strong biologic rationale for why the combination is needed and establishing the contribution of components, because what we don't want is a regimen that has 1 active agent and a toxic placebo. Getting into the dosage optimization of the combination regimen, some of the key considerations include, are there overlapping toxicities or PD interaction, PD interaction, which could lead to potentially increased toxicity? And also, is there the potential for PK interaction, meaning the presence of 1 drug affects the exposure of another? I want to come back to my point earlier about considering where the dosages of the individual agents optimized before being put in combination, because I can imagine a scenario where there aren't overlapping toxicities, there isn't a concern for PK interaction. However, the combination regimen as a whole would be very poorly tolerated because of the work that had been done on dosage optimization a priori or lack thereof. And in a situation like that, even if there's no concern for PK interaction, not a lot of concern for overlapping toxicities, further optimization of the combination regimen may be needed. And then finally, something else to consider when you're devising, how you're going to design the trials to support the combination are which agent contributes more to efficacy, which agent contributes more to toxicity, and that might help you decide which dosage combinations of the 2 or more agents to evaluate. One resource I want to point everybody towards is the co-development guidance, which FDA released in 2013. The purpose of this guidance was to provide recommendations for development of novel combinations, but a lot of the principles really apply more globally across combinations. And I've highlighted some of those principles on this slide. I won't talk too much about the ones that I've already covered on the previous slide, but some of the things that are in the co-development guidance are that strong biologic rationale for the combination is needed. It's important to understand the clinical safety and dose and exposure response relationships for the individual agents. Contribution of components needs to be established. And then evaluating multiple dosages of both drugs may help with optimization of the combination. And when you're considering how you will establish the contribution of components, which dosage of which agent to prioritize when you're deciding which combinations to evaluate, you can consider the anticipated or observed activity of the individual agents to guide you. 2 other resources I've just put on the bottom of the slide. FDA and ASCO co-sponsored a workshop in September 2023, so just a few months ago, on combination therapies in oncology, and some of the materials are available online. And then one paper that was discussed extensively at this workshop is this NCI paper with recommendations from a kind of a consensus recommendations from one of their groups regarding development of combinations. So I've put a link to that on the slide as well. Switching gears just for the last few minutes regarding dosage optimization of new indications. So I think there are 2 key overarching questions. The first is, was the dosage optimized for the initial indication? And then secondly, how does this new population that the drug is being developed for differ from the initial population? So some of those differences could include differences in tumor biology, differences in medical comorbidities. For example, the drug is associated with hypertension, and the new population, there are a lot of people with hypertension. Concomitant medication, so perhaps in one of either the new or the initial population, one of those groups, a lot of patients will receive antifungal treatments, but not the other, so something to consider. And then finally, the treatment setting. So maybe the drug was first developed in a late line metastatic setting. It's being moved up into earlier in the metastatic setting or even into the earlier adjuvant setting, and patients will be on therapy for much longer. In many of these situations, it's important to consider what is the benefit risk at the dosage that was established for the initial indication, and does that benefit risk change? And if so, how? If I put the same dosage into the new population, does further work need to be done or not to support that dosage? So in summary, both one of the commonalities between combinations and then new indications is that in both cases, there are existing data that can be leveraged to develop the combination, to develop the new indication. For combination regimens, factors affecting the need for additional dosage optimization include where the dosage is optimized for the individual agents, and then PK interaction and overlapping toxicities, PD interaction. And then for dosage optimization of the subsequent indications, consider the extent of dosage optimization for the initial indication and potential differences in the initial population and subsequent population. I think we'll talk about all of that in more detail throughout the day today. Thanks very much for your attention. And we would both like to thank all of the following people who had input on our presentation.
So I wanted to really thank our three presenters for bringing us a very nice presentation, which is important for us to set a stage for the subsequent discussions. And now the floor is open. I think if you have any questions, please come along to the microphone. And also for the audience who is attending virtually, you can use the chat box and type your questions, and we have our staff to help us to collect all the questions. Since I'm the session chair, I want to take a little bit of advantage for myself. Just want to ask some questions before we get some of our audience coming to the microphone. So the first question I actually have is probably to Dr. Chow. So how do you really factor in the patient population and the tumor heterogeneity into the dose selection? Let's say for if you were to consider something for general population.
It's a challenge, right? I don't think I have the answer for that. I just want to say that if you want to compare the dose, you better make sure the general population we refer have a reasonably comparable characteristics, tumor-wise, clinical, pathological-wise. Otherwise, the comparison will be unfair between the two doses. That's pretty much the current thoughts top of my mind.
Okay, thanks, Dr. Chow. So another question I have is for Dr. Thompson. I think you indicated that there are different types of messages that we need as from regulatory standpoint of view to consider dosage for a combination therapy. I'm just putting a little bit different scenarios. Let's assume we have two drugs. We have clinical information already for each of them as a monotherapy. Now we're moving to putting them together as a combination therapy. So what type of non-clinical information you would think it's valuable to make the dose selection in that scenario?
So FDA certainly operates in terms of this kind of multidisciplinary review team. So we're going to be clinical pharmacology. We're all going to be looking at it from our own way, from our own discipline. So I'd say our discipline, non-clinical, is really going to be focused on the safety of the combination. So thinking about overlapping toxicities, what was observed in the non-clinical studies that have previously been conducted. Because generally speaking, new non-clinical toxicology studies aren't warranted to support combinations, as long as they've been the products have been well characterized. So we're going to be considering overlapping toxicity based on the non-clinical data, potential for new safety signals, and maybe additional consideration from our point of view on what are the safety margins. So if you're going to propose two drugs in combination, are you going to to reduce maybe one of the dose levels on one of the drugs because of a concern for toxicity as you start to dose escalate the combination. So things like that. So really, it's a totality of the data approach where we're considering the pharmacology is important. It's important in terms of understanding the mechanism and understanding potential for new toxicities, but the toxicology is also very important in the consideration of the dosing.
Thanks very much, Dr. Thompson. Now I have another question for Dr. Shah. So let's assume that the sponsor is pursuing a new indication. They have some clinical experience for this compound with, let's say, monotherapy for using in one indication. They're making it into a new disease area. So what type of clinical information you would think it's important to consider? You know, can we actually use the existing dosing, or do we need to actually consider different doses? What that type of clinical information you would consider valuable? Thank you.
Yeah, I think that's the key question for the new indication. I can expand on some of the things I mentioned earlier, but it's really, you know, at the population level, how does the new population differ from the initial population? And do those differences, whether they're due to, you know, differences in tumor biology or comorbidities or certain medications that the patient population may be taking, do those differences lead to a reasonable hypothesis that a different dosage might have better benefit risk in the new population? Also, really, I can't emphasize enough my comments about considering, you know, what is the duration of therapy in the new population compared to the initial population? What is the anticipated benefit in that new population? How do those things balance? And does further work need to be done in terms of modifying, you know, not just the dose, but maybe the schedule and maybe the duration of therapy too? Because the fact is, most of our drugs are developed first in a late line kind of advanced cancer setting. There might be, even despite best efforts, there might only be observation of safety for a few months, you know, not necessarily on the order of years. And certain things might be tolerable for a few months that would not be tolerable in an early adjuvant setting if you're going to be on therapy for 2 years, 3 years. So those are some important clinical considerations.
Oh, thanks a lot, Dr. Shah. So maybe we can move to the audience. Maybe you can start from lady from my left side. You can just share your name and institute first, please.
Yes, I'm Helen Moore. I'm at the University of Florida, and it's wonderful to see all of these great speakers today and the focus on modeling and simulation in this area, figuring out combination therapies. So in the field, in the history, we've seen a lot of data in the many decades previously. We started using data, then we started using statistical methods beyond regression, and then we started using more powerful statistical methods like nonlinear mixed effects modeling. And now that we're using mechanistic models to answer questions like this, what are the optimal combination regimens? I'd like to get the panelists' perspective on what are your thoughts on bringing powerful methods from mathematics and engineering that have been developed for mechanistic models compared to where we are for the statistical methods. Have you given thought, or what are your thoughts? What are your responses to the idea that we might want to bring in more mathematical and engineering expertise in using these powerful methods they have?
Yeah, this is a really great question. Can I just start with Dr. Chow first?
All right, let me try give a step on that question. Great question. I think both valuable, like we did in our research. We're using mechanistic model to explore the situation, right? To see what is important, what is not. The statistical model equally important because that directly from the observation. It's not necessarily be mechanistically driven, but that's directly from the observation give us a top-down view about what is the important factor. We can integrate them together for sure to a large degree. You can see many publications on that topic. So to my knowledge, if you really want to get to the point to make decisions about approval or not, go or not go, those type of decision, I think a mechanistic model is important, but sometimes you have to give a whole health respect what the observation is. That's the statistical model stand out to become, more decision driven process. Is that fair to evaluate that situation?
I'll just mention yesterday at ACOP, we had an FDA approved device, a speaker who was talking about that device in diabetes, the artificial pancreas, and it relies on a mechanistic model because of the delay in the system. You have to have the insulin glucose model. So I was wondering if people are seeing that potential for more use of those methods.
Right. Another piece of information. In that case, depend on whether we have enough data to make a decision, right? If we don't have that enough data, then statistical model will not be feasible. We have to go back to rely on the mechanism we understand, knowledge we rely on. We collect it throughout the process. So it's really not a white black situation. It's really about what's the clinical scenario, what's the decision we want to make, how certainty we want to rely on those decisions. Again, combination probably is the future.
Thanks. I don't know if
I was actually going to ask what your opinion is.
my opinion. Okay, so maybe I can, if you wanted me to chime in, I think it's good to have different approaches. In my view, it's actually complementary to each other. Just a short answer to your question. Yeah. So I don't know if you have other follow-up questions. Okay.
I can make one comment.
Okay, please, go ahead.
Non-clinical perspective. So we see a range of models. Like I said, most of the models we see are going to be supporting the first in human dose selection right now, at least from our perspective, non-clinical perspective. So in terms of thinking ahead, any kind of modeling that can incorporate the different kinds of data types we see. So we see a range of biochemical, in vitro, and in vivo data across different species. So anything that can integrate that, and I'm not, whether it be PBPK or some other kind of modeling framework, could be whatever the sponsor wants to propose, as long as it's well justified and rational. But I would agree that we do need more mathematical and engineering backgrounds in the non-clinical discipline. That's my personal opinion on it. Most of our non-clinical staff are going to be pharmacologists or toxicologists, and we just need that more quantitative aspect in the reviewer in our reviewer portfolio. So.
Thank you. Maybe Dr. Gao, do you have some questions from online? We can. Okay, so maybe you can move to ours.
Thank you. My name is Sandhya Girish, and I'm from Gilead Sciences. Really nice summary of the whole field by the presenters today. Personally, I've spent almost a decade on TDM1 and Herceptin, and teasing out the confounding factors can be really, challenging. And I was really struck by the slide that you showed, Dr. Chow, on the heterogeneity and how some patients may benefit from a low dose and some may benefit from a high dose. So just as a thought and kind of to stimulate some, thinking, I was just wondering, from a patient perspective, would there be a willingness to perhaps look at 2 doses or approve 2 doses and give optionality to the patients so we can really tweak either the safety or the efficacy at the patient level? So I would like to hear, some thoughts around that from even the oncologist or the panelist perspective.
Dr. Chow, would you like to start?
Sure, I think that's a great question, and actually something that we hear a lot from patients. I think you're absolutely right that there is a lot of interest from patients in knowing, is this the right dosage for me? And some of that may be, is this the right dosage for me in terms of, what we can do from a regulatory perspective, which I'll address in a moment. And then also some of that might be also practice of medicine in the clinic. From a regulatory perspective, there's no reason why we couldn't approve 2 dosages to put in the label if there were strong rationale that both had favorable benefit risk. There were data to support both dosages, and then there were, considerations that could be labeled about why, certain patients might receive better benefit risk with one dosage versus the other. But there's no reason to prevent something like that if both dosages have demonstrated favorable benefit risk, and there's data to support approval of both of them.
Thank you very much. That's very helpful. No.
Yep. Thanks a lot. Maybe we can move to this side and Thanks. That's a good question. How do we want to organize the materials in a better format and so that could communicate easily? So I don't know who wants to take a shot first.
I can start and then see what my FDA colleagues have to add. So I think that's a great point. You know, there's something that we have now used for the past, I don't actually know how many years, but the past few years at least, which is a voluntary submission when an NDA or BLA package is coming in called the assessment aid. And that is one place where it is, you know, much more streamlined for us to see, you know, it's one of the reasons that the assessment aid was piloted was because reviewers were doing a lot of copy paste out of the submission and into their own review template. But the other really great thing from my perspective about the assessment aid is that it allows us to see your thinking and your interpretation of the data that you're presenting. And so we can say, you know, do we arrive at that same interpretation? Are there important considerations that we would add to, you know, to what you're saying? And that might change our interpretation. So I think generally that's a good document to do exactly what you say, you know, how do we put all this data together and interpret it? I think you raise an interesting point though, that it is still divided by disciplines. And is there, you know, opportunities for better crosstalk among disciplines? I think that's actually a common theme across dosage optimization is a lot more crosstalk between disciplines is required than maybe was traditionally done, especially in earlier development and, you know, how to capture all of that. But thanks for the question.
Yeah, it's a great question. And we do review as a team, but we tend to focus on to get technical, like we have modules that we look at. So if you submit something to the non-clinical module, then we're going to be reviewing that. If, for example, just going back to, I know this is not for, this is first in human here, but I mean, I love to see the modeling reports submitted into the non-clinical modules based on the non-clinical data. I think it's a very interesting kind of submission to look at when all the data is integrated. But yeah, it does address this when you get to, as you're developing the drug, and how are you bringing in the other review disciplines? A lot of times as you move forward, let's say, so you've done your initial dose selection, the non-clinical data tends to become less important, honestly, in terms of once you have that clinical data, that tends to be the driving force. So sometimes I've certainly seen cases where a drug has had issues in development because we saw severe toxicity, for example, in the non-clinical studies at exposures that are where at an exposure level that is actually being investigated as a recommended dose for follow-up. And it's like some people just aren't looking back towards the non-clinical data in a more integrated way. And so I think it's going to be important that we there was a great talk yesterday, and it was by somebody I think was involved in one of the PD-L1 development programs, and they talked about how a lot of the decision making as you get further in development was made by MDs, and how Project Optimus for them was an opportunity to pull in more of the pharmacometrics and more of the clinical pharmacology into the decision-making process. So I'd also say that as this area emerges, we also need to kind of think about how we can incorporate the non-clinical view more into the decision-making process as well.
Thanks a lot. Maybe we can pause a little bit from the in-person questions. We can go to the virtual attendance just to see if we have some questions from online. Thank you.
There's question online trying to understand when we are trying to discerning the causal dose response and exposure response relationship in early oncology trials. How important do you think the model-based approaches can help here?
Dr. Chow?
Let me give a try. I think the virtual control might be an important approach for us to leverage for the trial design, not probably for the internal decision making, probably not for the final part of the final analysis. But if we are doing the drug, development, there are many cases where compared to the same control, why not just share them or create a virtual one or enrich that control so that our power can be increased accordingly. But one thing I do want to point out, to create a virtual control, we should be able to understand what's the most important factor there. So that's the second step. We, the first step is to try to understand all those factors related to the response and toxicity. Then you can create a virtual control. So the virtual control is a good approach. Still conditioning on those understanding about all those factors related to the response and toxicity.
Thanks. Do you want to take one more from online or somebody?
I was just going to add a quick comment that, one of the challenges in that setting might be you might not know what the most important factors are if you're very early in development, So of course, one of the reasons we like things like randomization and tools like that is because it allows for potential balance between those unknown factors too, and not just the ones that you're aware of and can adjust for.
Yeah, we can take one more question from online.
Maybe we can take one more. So this one is specific to Dr. Shah, and the audience wants to understand a little bit the expectation for dosage optimization in the rare disease setting when the patient population may be quite low.
Yeah, so I think that's really a great question. And so I think that is a large reason why we have so much flexibility built into our recommendations, because we do want a dosage optimized for benefit risk for all populations that a new drug is being developed for, but we definitely understand that there are some feasibility limitations in a rare population. And so definitely encourage early meetings with us if you're developing a drug, and many oncologic indications are rarer, but there's some that are truly in very rare patient populations. So definitely encourage meeting with us early in that situation to discuss your strategy. And so we have an understanding of the limitations and we can provide recommendations.
Thank you. We're coming back to our in-person attendance room here. So maybe we can continue our question here. I think we can start from this side, then we can move to the middle. Yeah. So I think the question is, which you highlighted here, is some of the confounders actually will bias the potential readouts from the trial, where given the small sample size we're dealing with, right? Yep. And we should. Yeah, I think we should emphasize that, and maybe I can just turn it over to our Dr. Shah, so that you can get started first.
Sure. I think it's a really interesting question about, the importance of culture when making decisions. I will say that, across disciplines at the FDA, we bring a lot of humility to the things that we potentially do not know when we're looking at the data that's submitted to us. And I think we do our best to communicate, so what some of our uncertainties are when we're looking at things that have been submitted at milestone meetings throughout drug development. Want to just repeat my points from earlier too, and reflect on Dr. Thompson's points about the importance of kind of crosstalk between disciplines and having a lot of understanding of what somebody else might know that you might not know and vice versa. But yeah, I think that's really interesting, kind of the importance of culture and creating open-mindedness to make the best decisions going forward.
Thank you. Okay, thank you. So do you have? Okay, so maybe we can move to the middle here, Jean.
Jin from Genentech. I actually have two questions, one clinical, one preclinical, but I'll probably just choose one to ask. So I'll ask my clinical question. This is more of a question to Dr. Shah and maybe also for the panel. You have one slide to emphasize in the combo setting, it is important to characterize the dose for each single agent first. Now, for combo setting, we also have scenarios that based on MOA, there's no single agent activity expected. Then for those scenarios, it's arguable how much dose escalation is even ethical to do in patients for the agents without any hypothesis for single agent activity. What's your advice in those scenarios?
Yeah, so great question and something that's been under a lot of discussion. I would actually point you back towards the co-development guidance that I mentioned in my talk. There is a lot of discussion in there about, you know, if one of the agents is anticipated or observed to have limited single agent activity, you know, how do you get to the combination faster? How do you prioritize, you know, which dosages of the combination to evaluate? I will say though that even in that scenario, it's very important to have an understanding of the clinical safety of that individual agent, and also, you know, of course, establish the contribution of components, because we want to make sure that that agent that does not have monotherapy activity is actually enhancing the activity of the other agent or agents.
Thank you. Maybe I just want to repeat your question for people who are online. There's having some trouble to understand or to hear your question here. So I think the question is, I think Jean just presented a scenario where the combination therapy is supposed to be working, but each individual compound is not anticipated to be efficacious. Generally speaking, we wanted to look at the dose response for monotherapy first before we get into the combination scenario. However, for this situation, how we want to look at a dose, and given the understanding that they're not anticipated to be efficacious individually, is that right? Okay, thank you. So now we can move maybe for people to this side and just raise your question here.
Hi, good morning. Madhav from Vantage Research. So I was wondering, given the population variability even within a single indication, and you know the goal of not subjecting patients to unwanted toxicities, is there any idea of moving away from per weight dosing or per surface area dosing to per biomarker level based dosing?
Okay, so your question is more to say, can we actually use biomarker to drive dose selection rather than, you know, the traditional approach?
Yeah.
Okay.
Sure, I can just point you towards a clinical example, you know, not necessarily a blood-based biomarker, but if you look at the labeling for axitinib, you can see that they use safety endpoints, so things like hypertension and just more global safety events to titrate the dose of that product. And it's one example where FDA was able to label kind of a flexible dosing regimen in section 2 of the labeling because of the way that product was evaluated throughout its development, and kind of the way that a safety endpoint was used to guide dosing.
Thank you. So maybe I'll pause for a minute. We can just invite our virtual attendants to raise some questions too. Thank you.
Yeah, so there's a great question about to all the panelists. I think Dr. Shah, you mentioned about the combination rule and also the desire to understand the contribution of single agent. So this question wanted to hear the panel's insights on opportunities and challenges to inform the combination of components using model-based approaches in lieu of formal comparative clinical trials. So maybe we'll start with Carter.
Not quite sure I fully understand the question, but yeah, modeling and simulation or the model-informed approach certainly can help us to understand the contributing components to the combination right? That's the thing we are doing to understand what's the reason why the combination effective or tolerance. My short answer is yes, why not? We are using modern simulation all the time. Certainly the modern simulation can provide some complementary evidence for us to design the combination and from each drug or the final dose selections for each of these drugs in the combination.
Yeah, thanks. Thanks for that perspective. So yeah, I think the key word there though is complementary, because I think, very important to still have clinical demonstration of the contribution of components because of all of the factors that we've been talking out throughout this session regarding uncertainties about assumptions and differences between the patients who might have informed the modeling compared to patients that are receiving the drug in trials and would receive the drug in practice. Some of those things might not even be known. So I think complementary is really the key word, but still, still need contribution of components demonstrated and clinically.
Yeah, now we can come back to our room here. Maybe we can come back to this side and just again, if you have any questions, we can raise here.
I would say that we would want to see a characterization of the toxicity, the safety from a non-clinical perspective for the individual agents. But if you're going to start combining that many, there would not be an expectation that you conduct toxicology studies with 3 and 4 agents. Modeling, would be supportive in a way. If you want to present a model that integrates all the information, that could be supportive. But from our perspective, we'll certainly still be looking for individual characterization of the toxicity.
Great question. From a clinical side, I would say that it's pretty rare. I can't even think of a single example off the top of my head where it's truly a novel or novel combination. So I think most commonly when we're looking at 3 drugs, 4 drugs, 5 drugs, 2 of those drugs have already demonstrated a favorable benefit risk with contribution of components established, and 3rd agent is being established, being added, or there's a 3 or 4 drug chemotherapy regimen, which has demonstrated it's been standard of care for decades, perhaps for a particular disease, and a novel agent is being added. And so I would say that, in those cases, it's for contribution of components, it could really be standard of care versus standard of care plus the novel agent is probably most commonly what happens.
Yeah, just wanted to reiterate the question because it's just want to make sure that people who are calling in, they're understanding the question. The question is that what if we're looking at a combination for more than 2 drugs together? Thank you. So maybe we can move to the middle line here.
Thank you. This is Valerio Damian from GlaxoSmithKline. So my question is, looking back at the conference that we just had, there is a lot of discussion about mechanistic modeling as well as artificial intelligence and machine learning. So the question is, given the same context of use, the same kind of data, which of these 2 approaches carries a higher weight of evidence? And I can make the question a little bit harder, perhaps even more challenging. What if the 2 approaches propose 2 different doses? Which one would you choose?
Okay. So if I can reiterate your question, the question is that we have different modeling approaches, for example, machine learning, mechanistic modeling, and which one actually gives a higher weight given the same context of use? And what if we get different outcomes from different modeling approaches? So Dr. Chao, you wanted to get started?
I want to defer you to take.
You want to defer to me? Well, I have to say it really depends. I don't have a solid answer. It has to get into the specific context we're looking at, and to see. To me, I don't just by looking at on face, I don't see. We have to trust one model versus another model more. It's just a model, one of those approaches we're looking at. So we need to look at what data they use for modeling building, and what type of information they use for model validation, and what type of other pieces of information may help us to factor in the final decision. So I don't have a solid answer, but I think we have to look at all the pieces of information altogether.
I can provide a comment too. I'm a clinical person. I'm not a modeling expert, but usually when I'm asked to look at models, whether it's by my statistical colleagues, clinical pharmacology, pharmacometrics colleagues, it's to look at what the clinical assumptions were that went into that model. And so I would say, for any model, no matter how simple or how complicated, if they're proposing different results, then I would be looking pretty carefully at what the assumptions were that went into the modeling. And for me, it would be what clinical assumptions went into that modeling and how valid they are, what might be some of the gaps there.
Thank you. The question, if I may further this, it's around what's the value of having the model be mechanistic and being able to understand mechanistically what why you are proposing the dose that you are proposing, whereas a machine learning may actually tell you what it should be, but not necessarily provide you the justification.
Just a very brief comment on the machine learning versus the mechanistic model. So I remember years ago when I look at New York Times, the title, the page cover is telling us that the machine learning have a feature which is a data hungry, right? The machine learning, if you compare to the regression approach, is require 10 times for higher data value than the regression approach, meaning that if you do a linear regression, you probably need 3 points for regression, but machine learning need 30 data points to give you a reasonable prediction. That's a technical aspect I just want to add.
Thank you.
Yeah, if I can just to chime in one word is, I think even mechanistic model, you actually borrowed a lot of information from different sources. That adds a lot of uncertainty as well. So it's not like truly mechanistic. That means solid. Okay, maybe we can move on to this side and to see other questions from the floor.
René Bruno from Genentech. So the dose optimization question is really a complex one, as we probably all figure out. Do we want to optimize the dose at the population level, at the level of subpopulations of patients, or even at the patient level? And do we want to, and when we have a combination, the problem is multiplied. And probably to tease that out, we, from an experimental design point of view, we may need hundreds of experiments to look at all the combination in the different populations and all that. So I think we really need to think about the endpoints that we are going to use for that. Are we going to base that on the gold standard overall survival, or to accept to have intermediate endpoints like PFS or response rate? But those are not very informative. So are we open to do some optimization with, we have heard about biomarkers or longitudinal biomarkers like ctDNA, tumor dynamic models, to integrate all this information. So is it conceivable that within a treatment that has shown benefit in survival, for example, in a pivotal trial, to do optimization for subpopulations and even individual patients based on this type of endpoints and some models based approaches?
Okay, so maybe I can just reiterate your question. I think your reason, my view, is a very important point, is what is a target for what we call optimization? Do we talking about the general population or subgroups of populations, and can we use some of the biomarker to guide that type of decision making? Right. Okay.
I guess I can start, and then interested to see what my panel colleagues have to add. So it's actually thinking about something a little bit different. So of course, the efficacy endpoints that support a regulatory approval might be different than those that are used to look at activity throughout early development that make up the totality of data when optimizing the dosage. I could imagine a scenario where there's a particular PD marker that might be very helpful in early development for a particular product. Actually, I was thinking when you were talking, even if there are multiple combinations of drug A and drug B that you want to evaluate and you want to see, I predict that this regimen might have slightly better toxicity, but I want to see, could it be anticipated to have the same activity as this other combination? Something like that might be very helpful. And I think similarly, might be helpful to have early signs about specific populations within the general population that might need a different dosage or more support around which dosage to take forward for them.
Thank you. I think we have only 3 minutes left, so we can take one more question and we're going to close our section. So I'm sorry if there's other question has not been addressed. I would encourage you to talk to our panel members after the meeting, so we can take maybe lady first. So I think the question is about using healthy volunteers for some of the drugs which we consider appropriate to explore, to make a further exploration before we get into patients, right? Okay, so.
Sure, I can provide a response. So I think that's, a really interesting thought, interesting point, something that people are thinking about. I think it depends on, what do we mean by very little toxicity? Is it very little anticipated toxicity for an oncologic population versus would it be something that would have acceptable benefit risks to put into a healthy volunteer population? But I think that's a great point for, potential for certain products. There might be potential to evaluate them in healthy volunteers.
So I think we're getting at the right on time now. So I wanted to thank our presenters giving us such wonderful presentations, and also our panel members for sharing their thoughts. And also I wanted to thank our audience for your very great discussion. I think this is really a fruitful discussion, and I hope we can continue that discussion even after this, this workshop and the meetings. I think science is evolving, and I think we're going to probably evolving together. But the ultimate goal is how we can benefit our patients by introducing a very effective combination therapy, as well as targeting new indications. Thank you all.